Have you ever found yourself just, drowning in information, skimming headlines, maybe getting the gist of things, but not really getting it, not truly understanding?
Oh, absolutely. It's easy to feel like you're just scratching the surface.
Exactly. Especially for the things that are, well, incredibly complex, things happening constantly right inside us, often totally unseen, but absolutely vital.
hmm Like all the biochemical processes ticking away every second.
Right. Keeping us alive, keeping us functioning, and we barely give them a thought.
It's pretty amazing when you stop and think about it.
It really is. So today we're doing one of our deep dives. We're tackling something truly foundational, really remarkable, the whole world of protein metabolism.
And how our bodies handle nitrogen, which is a huge part of that.
Yes, and that critical pathway, the urea cycle.
Right. The detox system, essentially.
Our mission today is definitely to go way beyond the surface. We want to break it all down, really aiming for that, comprehensive elite level understanding.
Getting into the nitty-gritty.
Exactly. How the body manages proteins, building them, breaking them down, how it deals with nitrogen disposal, and crucially, what happens when these incredibly precise systems go wrong.
Which can have major consequences, as we'll see.
Definitely. We've pulled together some really detailed biochemical source material for this, the kind that lays out the foundational science. We want to really grasp the masterful engineering happening inside us.
Sounds like a plan. Where should we start?
Let's start with the basics, but maybe in a way people haven't thought about it. Protein turnover. What exactly is that? I like to picture the body as like a bustling city.
Okay, I like that analogy.
Yeah, constantly under construction, right. Tearing down old buildings, putting up new ones, all at the same time. That's kind of what's happening with our proteins, isn't it?
That's a great way to put it. It's this continuous, simultaneous process, degradation, and synthesis happening side by side in every cell.
It's everywhere, all the time.
Omnipresent, yeah. In pretty much all forms of life. It's a fundamental state of dynamic renewal.
And the scale of it is pretty mind-blowing.
It really is. Just think, in humans, we turn over something like 1 to 2% of our total body protein every single day.
Wow, 1 to 2% daily.
Yeah, and a big chunk of that is muscle protein. It's a very active process, but it's not uniform. That's the key thing.
Meaning different parts break down faster than others.
Exactly. The rates of protein degradation vary incredibly widely. Depends on the tissue, depends on the specific protein, depends on the body's needs at that moment.
You mentioned some examples where it's really high.
Yeah, tissues undergoing major structural changes. Like uterine tissue during pregnancy is a classic example. Massive remodeling going on there.
Makes sense.
Or think about skeletal muscle during starvation. The body starts breaking down muscle protein for energy. That's a high turnover state.
Okay.
And, maybe surprisingly, even something like a tadpole losing its tail during metamorphosis. That's rapid protein breakdown, driving that change.
A tadpole's tail, that's a great unexpected example. So how do scientists actually measure this difference in rates?
They use a concept called protein half-life, or T12. It's simply the time it takes for the concentration of a specific protein to drop by half.
Okay, half-life, like with radioactive decay, but for proteins.
Sort of, yeah. Same principle. And when you look at these half-lives, the variation is huge.
Sure.
Take liver proteins, for instance. Some might have a half-life under 30 minutes,
Gone in half an hour.
while others can last over 150 hours. That's, what, more than 6 days?
That's an incredible range. So some are really transient, just needed for a moment, while others are built to last.
Precisely. Your typical housekeeping enzymes, the ones doing the basic everyday jobs like glycolysis, breaking down sugar for energy, they tend to have longer half-lives.
Yeah.
Maybe over 100 hours.
Because the cell always needs them running.
Right. They provide stability. But then you have the key regulatory enzymes. These are the ones controlling specific pathways, maybe turning them up or down.
The decision makers?
Kind of, yeah. And they often have very short half-lives, sometimes as low as half an hour to maybe 2 hours.
Why so short?
Because that rapid turnover gives the cell very precise, very quick control. If you want to shut down a pathway fast, you need to get rid of the enzyme controlling it fast. A short half-life allows that.
I see. It's like having a responsive dimmer switch instead of just an on-off button that takes ages to flip.
Exactly. It allows for rapid adjustments. And this constant breakdown and rebuilding, this efficiency, it actually raises a really interesting question.
Which is?
Well, if we're constantly freeing up amino acids, the building blocks, why don't we store the excess ones? We store fat, we store glucose as glycogen. Why not amino acids?
That is a good question. It seems wasteful not to, given how important they are.
It does seem that way at first glance. But the body has a different strategy. About 75% of the amino acids that get liberated when proteins are broken down, they get immediately reused to build new proteins.
Ah, so it's a very efficient recycling system.
Incredibly efficient,
Yeah.
like a cellular circular economy.
Yeah.
But that other 25%, the excess, it's not stored. That's a fundamental rule.
No amino acid savings account then.
Nope. Unlike fats or carbs, any amino acids not immediately needed for building new proteins are rapidly degraded, broken down.
And what happens to the pieces?
Well, their carbon skeletons, the main structure minus the nitrogen part, get converted into these versatile molecules called amphibolic intermediates.
Amphibolic, meaning they can go different ways.
Exactly. They can be plugged into energy-producing pathways like the TCA cycle, or used as building blocks for other things the cell needs.
And the nitrogen. That's the key bit for our discussion today.
That's the key bit. That amino nitrogen in humans and many other animals gets converted into urea, a much less toxic compound. And then we excrete that urea in our urine. That's the body's main way of getting rid of excess nitrogen safely.
Okay, so we know proteins are constantly being broken down, but how? It can't just be random chaos inside the cell, right? You use the analogy of a demolition crew.
Right, a highly specialized one. It's not random at all. The dismantling is handled by enzymes called proteases and peptidases.
The demolition crew members.
Pretty much. Intracellular proteases start the job. They hydrolyze, meaning they use water to break the internal peptide bonds, the links holding the amino acid chain together. This chops large proteins into smaller pieces, called peptides.
Okay, so big proteins become smaller peptides? Then what?
Then, other enzymes take over to finish the job, breaking those peptides down into individual amino acids. You've got endopeptidases, which keep snipping internal bonds in the peptides.
More chopping.
Then you have enopeptidases, which nibble away amino acids one by one from one end of the peptide, the amino terminus.
Starting from one end.
And carboxypeptidases do the same thing, but from the other end, the carboxyl terminus.
So it's a systematic breakdown from both ends and the middle until you just have single amino acids left.
Exactly. Very methodical. Ensures complete recycling of the building blocks.
But how does the cell decide which protein gets this treatment? What flags a protein for demolition? It can't be just age, can it?
Not just age, although damaged or misfolded proteins are definitely targets. But there are specific signals, too. One well-studied signal is something called a PEST sequence.
Pest. Annoying.
Maybe to the protein. It stands for proline, P, glutamate, E, serine, S, and threonine, T. Short regions within a protein that are rich in these 4 amino acids.
And having a pest sequence means?
It often targets that protein for rapid degradation. It's like a built-in degrade me quickly tag for certain proteins, especially regulatory ones that need tight control.
Interesting. Are there different overall pathways for this degradation?
Yes. Broadly speaking, we can categorize them based on whether they need ATP, the cell's energy currency. There's an ATP-independent pathway first.
No energy needed for this one.
Correct. This primarily happens inside lysosomes. Think of lysosomes as the cell's recycling centers or maybe tiny stomachs.
Okay. What kind of stuff do lysosomes handle this way?
A good example is certain glycoproteins circulating in the blood. As they get older or slightly damaged, they might lose a specific sugar molecule, a sialic acid, from their carbohydrate chains.
They get slightly modified.
Right. They become asialoglycoproteins. And liver cells have special receptors on their surface that recognize these slightly altered proteins.
Like a quality control check.
Exactly. The receptor grabs the asialoglycoprotein, pulls it into the liver cell via endocytosis, and delivers it to a lysosome.
And inside the lysosome.
The lysosome's powerful proteases break it down. No extra ATP needed for that final breakdown step. This lysosomal pathway also handles other things, like proteins brought in from outside the cell, some membrane proteins, and certain long-lived intracellular proteins. It's a general cleanup system.
Okay, that makes sense for general cleanup. But you mentioned regulatory proteins with short half-lives and misfolded proteins. That sounds like it needs more targeted control.
You're absolutely right. And that's where the ATP and ubiquitin-dependent pathway comes in. This is a huge deal in cell biology.
Ubiquitin. I've heard that name. Sounds important.
It's incredibly important. This pathway is critical for degrading those regulatory proteins with short half-lives, and also for getting rid of abnormal or misfolded proteins that could gum up the works or cause disease.
And this happens where? Not in lysosomes.
No, this happens out in the cytosol, the main fluid compartment of the cell. And it absolutely requires ATP energy and the small protein tag called ubiquitin.
So tell us about ubiquitin. What makes it so special?
Well, for starters, it's tiny, only 76 amino acids long, and its name ubiquitin comes from ubiquitous because it's found in virtually all eukaryotic cells, yeast, plants, animals, us, everywhere.
Okay.
But what's truly mind-boggling is how conserved it is through evolution. Compare the ubiquitin sequence in yeast to the one in humans. Billions of years of evolution separate us, yet only 3 out of the 76 amino acids are different.
Only 3. That's astonishing.
It really is. It tells you this molecule is doing something absolutely fundamental and essential, so its structure has been incredibly preserved.
Wow. Okay, so how does this tiny ancient molecule actually work? How does it tag a protein?
It gets attached to the target protein through specific chemical bonds. These aren't the normal peptide bonds linking amino acids in the protein chain. They're special bonds formed between the tail end of ubiquitin and a specific amino acid, lysine, on the surface of the target protein.
So it's like sticking a label onto the protein.
A very specific label, yes.
Yeah.
And interestingly, even the identity of the amino acid at the very beginning, the N-terminus of the target protein, can influence whether or how readily it gets ubiquitinated. It's another layer of recognition.
So it's not a simple attachment. You mentioned enzymes are involved
Wait.
and ATP.
Right, it's a whole enzymatic cascade, a 3-step process involving enzymes called E1, E2, and E3.
E1, E2, E3. Easy to remember.
Hopefully. So E1 is the ubiquitin activating enzyme. It uses ATP energy to essentially prime a ubiquitin molecule, getting it ready for transfer. It forms a temporary high-energy bond with ubiquitin.
Okay, E1 activates ubiquitin using ATP. Step 1.
Step 2 involves E2, the ubiquitin conjugating enzyme or transferase. E2 takes the activated ubiquitin from E1. It's like passing the baton.
Okay, A1 to E2.
Then comes the crucial step, E3, the ubiquitin ligase. E3 is the matchmaker. It recognizes both the specific target protein and the E2 carrying ubiquitin. It then catalyzes the transfer of ubiquitin from E2 onto that specific lysine residue on the target protein.
Ah, so E3 provides the specificity. It picks the protein to be tagged.
Exactly. And this is key. While there are only a few types of E1 and maybe a few dozen types of E2 enzymes in humans, there are hundreds of different E3 ligases. Over 500, maybe even more.
500 E3s.
Yeah. This huge variety of E3s is what allows the cell to specifically target thousands of different proteins for degradation in response to countless different signals. Each E3 recognizes its own set of target proteins. It's an incredibly sophisticated system for ensuring the right proteins get tagged at the right time.
That level of specificity is amazing. Now, I think I remember reading that it's not usually just one ubiquitin molecule, it's often a chain.
That's correct. We need to distinguish between monoubiquitination and polyubiquitination.
Okay.
Monoubiquitination is the attachment of just a single ubiquitin molecule. This often happens to proteins embedded in cell membranes. Usually this single tag doesn't immediately signal destruction. Instead, it often acts as a signal to change the protein's location or its interactions with other proteins, maybe directing it towards lysosomal degradation eventually, but it's not the primary signal for the main protein shredder.
So one tag is more like a routing signal. What about polyubiquitination?
Polyubiquitination is the main signal for degradation by the proteasome, the shredder we'll talk about next. This involves attaching multiple ubiquitin molecules, typically 4 or more, onto the target protein. These additional ubiquitins are linked together in a chain, often attached to specific lysine residues like lys-48 or lys-63 on the previously attached ubiquitin.
So it's like adding extra flags, making the signal much stronger and unambiguous.
Precisely. A polyubiquitin chain, especially one linked via lys-48, is the canonical destroy me now signal recognized by the proteasome.
Okay, so the protein is now tagged with a polyubiquitin chain. What happens next? Where does the shredder come in? The proteasome.
Exactly. The proteasome is the cell's major protein degradation machine for ubiquitin-tagged proteins. It's another large, complex structure also found ubiquitously in eukaryotes.
What does it look like?
It's often described as a barrel or cylinder. It's made of multiple protein subunits arranged in stacked rings, forming a central channel or pore.
A barrel with a hole through the middle.
Kind of. And crucially, the active sites of the proteolytic enzymes, the parts that actually cut the protein bonds, are located inside this central pore, facing inwards.
Hidden away. Why?
To protect the rest of the cell. You don't want these powerful protein-cutting enzymes just floating around freely, chopping up essential proteins that haven't been tagged. They're sequestered inside the proteasome core.
Makes sense. So how does the polyubiquitinated protein get inside to be shredded?
The entry is tightly controlled. The proteasome has caps, or regulatory particles, often at both ends of the barrel. These caps recognize the polyubiquitin chain on the target protein.
They act like gatekeepers.
Exactly. They bind the tagged protein, unfold it, which requires ATP energy, and then thread the unfolded polypeptide chain into the central proteolytic core.
So it has to be unfolded first.
Yes, it has to be fed into that narrow channel.
Oh.
Once inside the core, the proteases chop the protein into small peptides, typically 7 to 9 amino acids long. These small peptides are then released from the proteasome and further broken down into individual amino acids by other peptidases in the cytosol, ready for recycling.
And the ubiquitin tags, are they destroyed too?
Good question. No, the ubiquitin molecules themselves are generally not degraded. As the protein is being unfolded and fed in, enzymes associated with the proteasome cap, called ubiquitinases or DUBs, cleave off the ubiquitin chain. The ubiquitin molecules are then released and can be reused to tag other proteins. It's a very economical system.
Wow. Recognition, tagging, unfolding, threading, chopping, recycling the tags.
Yeah.
That is incredibly complex and elegant. No wonder it got a Nobel Prize.
Absolutely. The 2004 Nobel Prize in Chemistry went to Aaron Sykenover, Aram Hershko, and Erwin Rose for discovering this ubiquitin-mediated protein degradation pathway. It was a paradigm shift in understanding how cells regulate themselves.
And you mentioned it's clinically relevant when the system messes up.
Hugely relevant.
Yeah?
Defects in genes encoding ubiquitin itself, or the E1, E2, or E3 enzymes, or the ubiquitinases, or even components of the proteasome, can lead to a variety of human diseases.
Like what?
Things like Angelman syndrome, certain forms of Parkinson's disease that start early, von Hippel-Lindau syndrome, which predisposes to cancer, and a type of congenital polycythemia, which affects red blood cell production. It's also implicated in cancer development and neurodegenerative diseases like Alzheimer's. It just highlights how absolutely critical this quality control and regulatory system is for maintaining health.
Okay, so we've seen protein turnover and the sophisticated machinery for degradation inside cells. But you mentioned earlier, this isn't just happening in isolation. There's communication between organs, right? Especially for handling the nitrogen.
Exactly. Here's where it gets really interesting. Looking at the bigger picture, the whole organism level. Maintaining steady levels of amino acids in your blood plasma, especially between meals, depends on this constant back and forth, this net exchange between different tissues.
A metabolic conversation between organs.
That's a good way to put it. And two organs are really the star players in this conversation: muscle and the liver.
Muscle and liver, how so?
Muscle is a huge reservoir. It generates more than half of the entire body's pool of free amino acids, mainly from the normal turnover of muscle protein. So it's a major source, releasing amino acids into circulation.
Okay, muscle is the supplier.
And the liver. It's the primary processing plant, especially for nitrogen disposal. It's where most of the enzymes for the urea cycle are located, so it takes up amino acids from the blood to handle that excess nitrogen.
So muscle releases, liver takes up and processes. What does this flow look like, say, in the post-absorptive state, like a few hours after eating?
In that state, when you're not actively absorbing nutrients, muscle releases quite a bit of alanine and glutamine into the blood.
Alanine and glutamine specifically.
Yes, those are major carriers of nitrogen away from muscle. Alanine is primarily picked up by the liver. The liver can then use its carbon skeleton to make glucose, a process called gluconeogenesis.
Ah, making new glucose. Important when you're not eating.
Very important. Glutamine, on the other hand, is taken up mainly by the gut and the kidneys. Both can convert some of that glutamine into alanine, which might then go to the liver. Plus, the kidney uses glutamine for another crucial job. It can release the nitrogen from glutamine as ammonia directly into the urine. This is really important for maintaining the body's acid-base balance.
So glutamine has roles in nitrogen transport and acid-base balance.
Correct. And there are other movements too. The kidney also releases serine into the blood, which other tissues pick up. And those branch-chain amino acids, the BCAAs, like choline, muscle releases them, and they are predominantly taken up by the brain, especially during fasting.
The brain uses BCAAs.
Yes, they can be an energy source for the brain when glucose is less available.
You mentioned alanine going to the liver for gluconeogenesis. This sounds like the glucose-alanine cycle.
Exactly, you got it. The glucose-alanine cycle is a fantastic example of this interorgan cooperation. Alanine is a prime substrate for making glucose in the liver.
More so than other amino acids.
Much more so. The liver's capacity for making glucose from alanine is enormous. It doesn't even max out until alanine levels in the blood are like 20 or 30 times higher than normal.
Wow, huge capacity. So walk us through the cycle.
Okay. It starts in the muscle. Muscle breaks down glucose for energy, producing pyruvate. Through a process called transamination, muscle can add an amino group, nitrogen, to pyruvate, turning it into alanine.
Got it. Pyruvate becomes alanine in muscle.
Right. This alanine is then released into the bloodstream, travels to the liver, and the liver takes it up. Inside the liver, the process reverses. The amino group is removed. We'll see where it goes later, urea cycle. And the remaining carbon skeleton, essentially pyruvate again, is used to synthesize glucose.
Liver makes glucose from the alanine skeleton.
Yes. That newly made glucose is then released back into the bloodstream. Muscle can then take up that glucose, use it for energy, and potentially generate more pyruvate to make more alanine, completing the cycle.
So muscle sends nitrogen as alanine to the liver, and the liver sends energy as glucose back to the muscle.
That's the essence of it. It's a brilliant way to keep blood glucose levels stable during fasting or exercise, while also safely transporting potentially toxic nitrogen from muscle, which can't process it into urea, to the liver, which can.
A win-win for energy and detox. Now, what about after you eat a big protein meal? Does the flow change?
It does shift, yes. After a protein-rich meal, the gut absorbs the amino acids and they enter the portal vein, heading straight for the liver. These are called the splanchnic tissues releasing amino acids.
Okay.
Now, the liver takes up a good portion of these incoming amino acids, but it lets a significant amount pass through, especially the branched-chain amino acids, leucine, isoleucine, and valine. About 20% of the amino acids leaving the gut-liver area are BCAAs.
So the liver doesn't grab all the BCAAs right away.
Right, it sort of spares them. And where do they go? Primarily to peripheral muscle. Muscle eagerly takes up amino acids after a meal, and BCAAs make up a huge proportion of that uptake, maybe 60%.
So muscle is the main destination for BCAAs after a meal, while the brain was the main user during fasting. Why this special treatment for BCAAs?
It seems to be partly because muscle can readily use BCAAs for its own protein synthesis and also as fuel. By letting the BCAAs bypass the liver initially, the body avoids overwhelming the liver's nitrogen disposal system immediately after a large protein load. It distributes the processing load, with muscle handling a significant chunk of the incoming BCAAs.
That makes sense, sort of spreading the metabolic work. Now, we've focused on humans making urea, but not all animals do that, right? Fish, birds, they handle nitrogen differently.
Absolutely. It's a fantastic example of adaptation to environment and physiology. How an animal gets rid of excess nitrogen depends heavily on its lifestyle, particularly its access to water.
Okay, so fish.
Most fish live in water, obviously, so they can afford to excrete nitrogen in its simplest but also most toxic form, ammonia, NH3. They are ammonotelic. Ammonia is highly soluble in water and diffuses readily across their gills into the surrounding water, where it's quickly diluted to non-toxic levels.
So constant flushing with water makes ammonia excretion safe for them, but birds can't do that.
Definitely not.
Sure.
Birds need to conserve water, and they also need to be lightweight for flight. Ammonia excretion requires a lot of water, and ammonia itself is toxic if stored. So birds are uricotelic.
Uricotelic, meaning they make
Uric acid.
Yeah.
It's a much more complex molecule than ammonia or urea, very rich in nitrogen, but it's also much less toxic and, crucially, not very soluble in water. Birds excrete it as a semi-solid paste, guano. This gets rid of a lot of nitrogen with very minimal water loss.
clever solution for flight and water conservation. And then there's us.
And most other terrestrial mammals, yes. We are ureotelic. We convert excess nitrogen into urea.
Why urea?
Urea is a good compromise. It's far less toxic than ammonia, so we can tolerate relatively high concentrations in our blood without immediate harm. And it's highly soluble in water, so we can excrete it efficiently via urine through our kidneys, although it does require more water than uric acid excretion.
So urea is less toxic than ammonia, more soluble than uric acid?
Exactly. It's the Goldilocks solution for many land animals. And this low toxicity is important clinically. If someone has kidney failure, their blood urea nitrogen, BUN, levels go up. But the high urea itself isn't the primary cause of their illness. It's a consequence of the kidneys failing to excrete it. The real problems in kidney failure come from imbalances in electrolytes, fluids, and the buildup of other more toxic waste products, not primarily the urea itself.
Okay, so urea is our solution. And the process that makes it is the urea cycle, happening mainly in the liver. This is the core of our ammonia detox system. Can you give us the big picture overview?
Right. The urea cycle is absolutely central. If you look at the overall flow of nitrogen in the body, amino acids come in, their nitrogen needs to be handled, and the liver converts that nitrogen into urea for excretion. It's a hub.
And it happens in distinct stages.
Yes, we can break down the whole process of getting nitrogen from amino acids into urea into 4 major stages. First, transamination, gathering the nitrogen. Second, oxidative deamination, releasing the nitrogen as ammonia, mainly from glutamate. Third, ammonia transport, safely moving that ammonia. And fourth, the actual reactions of the urea cycle itself, where urea is synthesized.
Transamination, deamination, transport, cycle reactions.
Got it. And it's a highly regulated process. For instance, during starvation, when the body breaks down more protein for energy, the liver actually ramps up the production of the urea cycle enzymes. The genes for these enzymes get expressed more.
To handle the extra nitrogen load from protein breakdown.
Precisely. The liver anticipates the increased need to detoxify ammonia and prepares itself. It's an adaptive response.
Let's break down those stages. Stage 1, transamination. You said this is about gathering the nitrogen.
Exactly. Think of it as funneling. Most amino acids can transfer their alpha-amino group, their main nitrogen group, onto a molecule called alpha-ketoglutarate.
Alpha-ketoglutarate. That sounds familiar.
Yeah.
From the TCA cycle?
Yes, it's an intermediate in the TCA cycle too. Very versatile molecule. When alpha-ketoglutarate accepts an amino group, it becomes the amino acid glutamate.
So various amino acids give their nitrogen to alpha-ketoglutarate, turning it into glutamate.
Right. This is done by enzymes called aminotransferases or transaminases. These reactions are generally reversible, so they can also be used to synthesize amino acids when needed. Nearly all common amino acids participate, except for a few like lysine and threonine.
So it's a way to collect nitrogen from many different sources onto one molecule, glutamate.
That's the key outcome. Why glutamate? Because, as we'll see in the next step, L-glutamate is unique. It's essentially the only amino acid that undergoes oxidative deamination at a significant rate in our tissues.
So concentrating the nitrogen onto glutamate sets it up for the next step, the actual release of ammonia?
Precisely. All roads lead to glutamate, metabolically speaking, for nitrogen destined for urea.
You mentioned aminotransferases. Are they important clinically?
Very much so. 2 common ones are ALT, alanine aminotransferase, and AST, aspartate aminotransferase. If tissues rich in these enzymes, especially the liver, get damaged, the enzymes leak out into the bloodstream. So measuring elevated ALT and AST levels in blood is a standard part of liver function tests.
Okay. And these enzymes need help, right? Something with vitamin B6?
Yes. All aminotransferases require a cofactor derived from vitamin B6 called pyridoxal phosphate, or PLP. PLP acts as the actual carrier of the amino group during the reaction. It temporarily holds onto the nitrogen as it's transferred from the amino acid to alpha-ketoglutarate. It's essential for the reaction mechanism, which is often described as a ping-pong mechanism because substrates bind and products leave one at a time.
Fascinating. Okay, so stage 1 gathers nitrogen onto glutamate. Stage 2, oxidative deamination, releasing the ammonia from glutamate.
Correct. This is where the enzyme L-glutamate dehydrogenase, or GDH, comes in. It's found mainly in the mitochondria of liver cells.
GDH, what does it do?
It removes the amino group from glutamate, releasing it as free ammonium ion, NH4+, and in the process, it regenerates alpha-ketoglutarate, which can then accept another amino group in a transamination reaction.
So it releases the ammonia and recycles the alpha-ketoglutarate carrier.
Exactly. GDH is unique because it can use either NAD+ or NADP+ as its coenzyme for the oxidation part of the reaction. The combination of transamination, funneling N to glutamate, and GDH action, releasing N from glutamate as ammonia, is sometimes called transdeamination.
Transdeamination. Nice term. Is GDH regulated? It seems like a key control point.
It is. It's allosterically regulated. High-energy signals in the cell, like ATP and GTP, inhibit GDH. Makes sense if the cell has plenty of energy, it doesn't need to break down amino acids for fuel as much, so ammonia production can slow down.
And low energy signals.
ADP, a sign of lower energy, activates GDH. This links nitrogen metabolism directly to the cell's energy status. And like transamination, the GDH reaction is reversible, so it can also function to incorporate ammonia into alpha-ketoglutarate to make glutamate, playing a role in amino acid synthesis too.
Are there other minor ways ammonia gets produced?
Yes, there are L and D amino acid oxidases, mainly in liver and kidney. They directly oxidize amino acids, releasing ammonia and forming hydrogen peroxide, which is then quickly neutralized by catalase.
Mhm.
But GDH is really the major route for ammonia production from amino acid nitrogen in the liver.
Okay. Now, this ammonia, we established it's toxic, especially to the brain. How does the body handle it, even transiently?
That's a critical issue. Ammonia intoxication is serious. Normally, the liver is incredibly efficient at clearing ammonia from the blood coming from the gut, where bacteria produce some,
Mhm.
and from other tissues. It rapidly converts it to urea, keeping blood levels very low, maybe 10-20 OD shell.
But if the liver isn't working well, like in cirrhosis,
Then ammonia levels can rise dramatically in the systemic circulation. This happens in severe liver disease, or if blood flow bypasses the liver through abnormal connections. That's when you see the neurological symptoms: tremor, slurred speech, confusion, blurred vision, eventually coma and death if untreated.
and the brain toxicity mechanism, you mentioned it involves alpha-ketoglutarate.
Yes, the prevailing hypothesis is that excess ammonia crosses into the brain. Inside brain cells, it reacts with alpha-ketoglutarate, catalyzed by GDH actually running in reverse to form glutamate.
Consuming alpha-ketoglutarate.
Right. This depletes the brain's pool of alpha-ketoglutarate, which is a vital intermediate in the TCA cycle, the main energy-generating pathway. By draining this intermediate, ammonia impairs the brain's ability to produce ATP energy, leading to neuronal dysfunction. It might also involve messing with neurotransmitters like glutamate and GABA.
A metabolic disruption in the brain. Okay, so stage 3 must be about preventing this safely transporting ammonia.
Exactly. Since free ammonia is dangerous, the body primarily transports it between tissues in a non-toxic form, mainly as the amino acid glutamine,
Ah, glutamine again. How is it made?
by the enzyme glutamine synthetase. This enzyme is found in many tissues, including muscle and brain, and also in the liver mitochondria. It takes glutamate, adds ammonia to it, and uses ATP energy to form glutamine.
So it fixes ammonia onto glutamate to make glutamine.
Precisely. The reaction strongly favors glutamine synthesis because it's coupled to ATP hydrolysis, making it essentially irreversible under physiological conditions. Glutamine then serves as a safe, non-toxic carrier of nitrogen and carbon in the bloodstream, moving nitrogen from peripheral tissues primarily to the liver and kidneys.
You mentioned its role is vital.
Absolutely essential. There are rare, devastating genetic deficiencies of glutamine synthetase in newborns that cause severe brain damage and multi-organ failure. It underscores how critical this safe ammonia trapping and transport is.
So glutamine carries the ammonia safely. But eventually, the nitrogen needs to be released again, right? Especially in the liver for the urea cycle.
Correct. That's the job of the enzyme glutaminase. This enzyme does the reverse reaction. It hydrolyzes glutamine, releasing the amide nitrogen as free ammonia and regenerating glutamate.
Glutamine synthetase builds glutamine, glutaminase breaks it down.
Exactly. There are different forms or isoforms of glutaminase, a liver type and a kidney type, encoded by different genes and regulated differently. Liver glutaminase activity increases with high protein intake, providing more ammonia for urea synthesis. Kidney glutaminase activity increases during metabolic acidosis, releasing ammonia into the urine to buffer excess acid and help regulate pH.
So releasing ammonia in the kidney helps with acid-base balance.
Yes, it's a meteor mechanism. The kidney generates ammonia, mostly from glutamine, and secretes it into the tubular fluid where it buffers protons, H+, and gets excreted as ammonium ion, NH4+. This helps conserve bicarbonate, the body's main buffer, and gets rid of excess acid. Ammonia production goes up in acidosis and down in alkalosis.
Fascinating interplay. Okay, so we've gathered nitrogen transamination, released it as ammonia, deamination via GDH, and transported it safely as glutamine, then released by glutaminase. Now finally, stage 4, the urea cycle reactions themselves.
Right, the grand finale of nitrogen disposal in ureotelic organisms. This happens primarily in the liver. Urea is the end product.
And it's energetically expensive.
Very.
Yeah.
Making one molecule of urea costs the equivalent of 4 high-energy phosphate bonds. 3 ATP molecules are hydrolyzed to 2 ADP, 1 AMP, and pyrophosphate, which is then also hydrolyzed. The inputs are 1 molecule of ammonium ion, 1 molecule of CO2 as bicarbonate, and the amino group from 1 molecule of aspartate.
Ammonium, CO2, aspartate, and ATP go in, urea comes out. What are the key players in the cycle itself?
There are 5 core enzymatic reactions in the cycle and 6 amino acids involved in various ways. N-acetylglutamate acts purely as an activator. The others, ornithine, citrulline, arginosuccinate, and arginine, act as carriers for the atoms that eventually become urea. They get regenerated, so they're not consumed overall.
A true cycle, regenerating its carriers. Where do these reactions happen?
This is a key feature. Compartmentalization. The first 2 reactions happen inside the mitochondrial matrix. The remaining 3 happen out in the cytosol.
Mitochondria and cytosol working together, so molecules have to move between them.
Yes, CO2, ammonia, and ornithine need to get into the mitochondria. Citrulline needs to get out. There are specific transporter proteins in the inner mitochondrial membrane that facilitate this movement, like ORC1 and ORC2 for ornithine-acetylcholine exchange. This shuttling is essential.
Okay, let's walk through the 5 reactions. Reaction 1, the starting point.
Reaction 1 is catalyzed by carbamoyl phosphate synthetase I, or CPSI. This is the mitochondrial matrix. It takes CO2 as bicarbonate, ammonia, NH4+, and 2 ATP molecules and combines them to form carbamoyl phosphate.
Carbamoyl phosphate, that sounds like a high-energy molecule.
It is. It has a high group transfer potential. CPSI is distinct from CPS2, which is cytosolic and involved in making pyrimidines. CPSI is dedicated to the urea cycle. It's a rate-limiting step, the pacemaker for the whole cycle.
And it needs that activator you mentioned, N-acetylglutamate.
Yes, CPSI is essentially inactive without its allosteric activator, N-acetylglutamate.
Yeah.
And acetylglutamate binds to CPSI and increases its affinity for ATP, turning it on. And remember, this step uses 2 ATPs per carbamoyl phosphate formed. One activates bicarbonate, the other drives the synthesis of the imide bond. Very energy-intensive start.
2 ATPs just to get started. Okay, reaction 2. What happens to carbamoyl phosphate?
Still in the mitochondria, ornithine transcarbamoylase, OTC, takes over. It transfers the carbamoyl group from carbamoyl phosphate onto ornithine. Ornithine is one of the key carrier amino acids. The product is citrulline, and phosphate is released.
So carbamoyl group plus ornithine equals citrulline. Still in the mitochondria.
Right, but now citrulline needs to get out into the cytosol for the next steps. So it's transported out via that mitochondrial carrier, often in exchange for ornithine coming in.
Okay, citrulline is now in the cytosol, reaction 3.
In the cytosol, arginosuccinate synthetase combines citrulline with the amino acid aspartate. This reaction requires ATP, which is cleaved to AMP and pyrophosphate, equivalent to using 2 high-energy bonds. This step is crucial because aspartate provides the second nitrogen atom that will end up in urea. The product is arginosuccinate.
So citrulline plus aspartate plus ATP arginosuccinate plus AMP plus PPI. Now we have both nitrogens incorporated into arginosuccinate.
Exactly. Reaction 4 is next, still in the cytosol. Arginosuccinate cleaves arginosuccinate into 2 molecules, arginine and fumarate.
Arginine and fumarate.
Right. The arginine molecule now contains both nitrogen atoms destined for urea. The fumarate released is the carbon skeleton of the aspartate that came in during the previous step.
What happens to the fumarate? Is it wasted?
Not at all. Fumarate is an intermediate of the TCA cycle. It can be converted via cytosolic enzymes, fumarase, malodehydrogenase, to oxaloacetate. This oxaloacetate can then be transaminated, using glutamate, back into aspartate. So the carbon skeleton is recycled to bring more nitrogen into the cycle. It's another link between the urea cycle and general metabolism.
Very efficient recycling loop. Okay, we have arginine with the 2 nitrogens, reaction 5, the final step.
The final step, also in the cytosol, is catalyzed by arginase. This enzyme hydrolyzes arginine, splitting off urea, which contains the 2 nitrogen atoms. The other product is ornithine.
Urea is released, and ornithine is regenerated.
Exactly. That ornithine can then be transported back into the mitochondria to pick up another carbamoyl phosphate, starting the cycle all over again.
A perfect cycle. Urea goes off to the kidneys for excretion. Ornithine goes back for another round. Amazing. How is this whole thing regulated? You mentioned CPSI is the pacemaker.
Yes. CPSI activity, dictated by the levels of its activator, N-acetylglutamate, is the primary short-term regulator. The level of N-acetylglutamate itself depends on the balance between its synthesis by N-acetylglutamate synthetase, NACS, using glutamate N-acetyl-CoA, and its breakdown. Factors like arginine levels can stimulate NACS, linking cycle activity to substrate availability.
And longer-term regulation, like during starvation.
That involves changes in the amount of the enzymes. As we said, during starvation or on a very high-protein diet, the liver increases the synthesis of all 5 urea cycle enzymes, sometimes 10 to 20-fold. This is long-term adaptation to handle sustained high loads of nitrogen.
It's such a complex, coordinated process, which means Unfortunately, there are multiple points where things can go wrong due to genetic defects. These urea cycle disorders sound incredibly serious.
They are. They're relatively rare individually, but collectively they represent a significant group of inherited metabolic diseases, and they can be medically devastating, especially if not diagnosed and treated quickly.
Studying them must teach us a lot about metabolism in general, though.
Absolutely. They beautifully illustrate several key principles that apply to many inherited metabolic diseases.
Such as?
Well, first, you can get very similar, sometimes identical clinical signs and symptoms, even if the genetic defect is in different enzymes within the same pathway, or even different mutations in the same gene. Makes diagnosis tricky based on symptoms alone.
Okay, same symptoms, different root cause sometimes.
Second, developing rational treatments absolutely depends on understanding the underlying biochemistry, knowing which reaction is blocked, what's building up, what's deficient.
Makes sense. Need to know the chemistry to fix the chemistry.
Third, identifying what builds up before the block is key for diagnosis. Those accumulating intermediates become the biomarkers we look for in screening tests.
Like checking for high citrulline if the block is after citrulline formation.
Exactly. Fourth, definitive diagnosis often requires directly measuring the activity of the suspected deficient enzyme in patient cells or tissues.
To confirm it's not working properly.
Fifth, genetic testing. Sequencing the DNA of the relevant gene can pinpoint the specific mutations responsible.
Getting down to the precise error in the code.
And finally, thanks to widespread sequencing, we now know there can be many different mutations within the same gene, leading to a whole spectrum of severity from relatively mild issues to severe life-threatening disease. Huge variability.
So what are the common threads among the urea cycle disorders themselves?
There are 5 well-characterized disorders, one for each of the core enzymes: CPSI, OTC, arginine-succinate synthetase, arginine-succinate arginase. Despite the genetic variability, they all tend to share some core clinical features.
Which are driven by the ammonia buildup.
Primarily, yes. Hyperammonemia, high blood ammonia, is the hallmark. This leads to encephalopathy, brain dysfunction. Patients often also have respiratory alkalosis.
And the substances before the block accumulate.
Right. In defects of the first 4 enzymes, you see accumulation of urea precursors, especially ammonia itself, and also glutamine, as the body trying to trap the excess ammonia. The earlier the block in the cycle, like CPSI or OTC deficiency, generally the more severe the hyperammonemia because less ammonia gets processed at all.
And the symptoms this causes.
Symptoms can include vomiting, particularly after protein intake, leading to an aversion to high-protein foods. Also, intermittent ataxia, poor coordination, irritability, lethargy, and if untreated, severe developmental delay and intellectual disability due to the toxic effects of ammonia on the developing brain.
It sounds particularly dangerous for newborns.
It is. Often an infant with a severe defect might seem perfectly normal at birth, having been protected by the mother's metabolism. But within days, as they rely on their own urea cycle, ammonia levels skyrocket, leading to rapid onset of lethargy, poor feeding, temperature instability, hypothermia, and episodes of apnea. Stop breathing. It's a true metabolic emergency.
Terrifying. How are these managed? Is there treatment?
Yes, and early intervention is key. The treatment principles are similar across the different defects. The cornerstone is dietary management, a severely protein-restricted diet.
Low protein to reduce the nitrogen load.
Exactly, but it's a very careful balancing act. You need to provide enough essential amino acids and calories for growth and development, but not so much protein that ammonia levels become toxic. Often involves frequent small meals, special low-protein formulas, and sometimes supplementation with essential amino acids or arginine, depending on the defect.
A lifelong dietary challenge.
For many, yes. The goal is to minimize ammonia spikes and allow for the best possible neurological outcome. Sometimes medications that help scavenge nitrogen are also used.
Let's quickly run through the specific disorders. CPSI deficiency.
Deficiency of carbamoyl phosphate synthetase I, reaction 1, causes hyperammonemia type 1. Relatively rare. Remember, N-acetylglutamate is essential for its activity.
And the disorder related to that activator, NEGS deficiency.
Right. N-acetylglutamate synthetase deficiency causes symptoms almost identical to CPSI deficiency because the activator isn't being made. Diagnosis can be tricky, but the potential good news is that some NEGS deficient patients respond well to being given N-acetylglutamate itself as medication.
A very targeted therapy if it works. What about getting ornithine into the mitochondria?
That's ornithine permease deficiency, or HHH syndrome, hyperornithinemia, hyperammonemia, homocystinuria. A defect in the ORC1 transporter means ornithine can't get into mitochondria, shutting down the cycle. Causes high ammonia, high ornithine in the blood, and a unique marker. Homocitrulline in the urine, formed when carbamoyl phosphate reacts with lysine instead of ornithine.
Okay, OTC deficiency. Reaction 2.
Ornithine transcarbamylase deficiency, hyperammonemia type 2. This is the most common urea cycle disorder, and it's X-linked.
Meaning it affects boys more severely?
Yes, typically. But female carriers can also have symptoms, like hyperammonemia or avoiding protein. High glutamine levels are often seen in blood and urine, reflecting the body's attempt to buffer the ammonia.
Next, arginosuccinate synthetase deficiency. Reaction 3.
Also known as citrullinemia. The block here leads to a massive buildup of citrulline in the blood, cerebrospinal fluid, and urine, sometimes grams excreted per day. The enzyme might be missing entirely or just have very poor function.
And arginosuccinate lyase deficiency. Reaction 4.
That's arginosuccinic aciduria. Here, arginosuccinate itself accumulates significantly. Some patients with this disorder have a distinctive feature, brittle, tufted hair called trichorexis nodosa. There are early and later onset forms. Enzyme activity can be measured in red blood cells or even prenatally.
Finally, arginase deficiency, the last step, reaction 5.
Yes, hyperarginemia. This one often presents a bit later, maybe age 2 to 4, not always in the newborn period. Arginine levels become very high in blood and CSF. Interestingly, the high arginine competes with other amino acids like lysine and cysteine for reabsorption in the kidney, so you can see a pattern in the urine that mimics a different disorder called lysine cystinuria.
These are clearly devastating conditions, but the fact that we can diagnose them and manage them with diet is huge, especially with newborn screening. That seems like a massive public health success story.
It absolutely is. Early detection through newborn screening is probably one of the most impactful advances in pediatrics and inherited metabolic diseases. Finding these conditions within days of birth, before severe, irreversible brain damage occurs, can completely change a child's life trajectory.
How did screening evolve?
It started back in the 1960s, initially just for PKU, phenylketonuria, but now, thanks largely to a technology called tandem mass spectrometry, or tandem MS, screening has expanded dramatically.
Tandem MS, how does that work?
It's an incredibly sensitive analytical technique. From just a tiny spot of dried blood collected from a baby's heel prick, tandem MS can measure the levels of dozens of different metabolites, amino acids, organic acids, acylcarnitine simultaneously and very quickly in just minutes.
Wow, dozens of tests from one blood spot.
Exactly. Most states now use tandem MS to screen for a whole panel of disorders, including amino acidopathies, organic acidemias, fatty acid oxidation defects, and, relevant to our discussion, several urea cycle disorders by looking for those telltale accumulating intermediates.
That's amazing. A real game changer for catching these early.
Truly, it allows for diagnosis and initiation of treatment, usually dietary changes, often before the baby even shows any symptoms. It has saved countless lives and prevented untold neurological damage.
So detection and management have improved dramatically. What about cures? Fixing the underlying genetic problem? Is gene therapy an option?
That's the ultimate goal, of course. There has been research and some success in animal models using viral vectors to deliver a correct copy of the gene, for example, in models of citrullinemia, arginosuccinate synthetase deficiency. However, translating this safely and effectively to human patients for urea cycle disorders has proven challenging. Right now, gene therapy isn't a standard clinical solution for these conditions.
So not quite there yet for humans. Any other approaches showing promise, like CRISPR?
Yes. The gene editing technologies, particularly CRISPR-Cas9, offer exciting possibilities. Research has shown that it is possible to use CRISPR to directly correct the faulty gene in cultured human cells derived from patients, specifically in pluripotent stem cells.
Correcting the gene in patient-derived stem cells.
Right. They were able to restore functional enzyme activity in the lab. Now, this is still a long way from a therapy you can give to a person. There are huge challenges regarding delivery, efficiency, safety, and off-target effects, but it's a proof of principle. It shows that precise genetic correction might be feasible down the road.
So while not a clinical reality today, the potential to actually fix the root cause using tools like CRISPR is definitely on the horizon.
It is. It represents a potential paradigm shift from lifelong management to perhaps a one-time corrective therapy in the future. But there's still a lot of research and development needed to get there safely. #tag #AdCheckoutTriddle.
Wow. What an absolutely incredible deep dive. We've journeyed from the constant dynamic turnover of proteins within every cell through the sophisticated tagging and disposal systems like ubiquitin and the proteasome, and followed nitrogen's path through transamination, deamination, safe transport, and finally, the intricate life-saving reactions of the urea cycle.
It really highlights the sheer complexity and honestly the elegance of our biochemistry. Every step, every enzyme, every transporter meticulously coordinated to maintain balance and detoxify waste.
And we've seen how critical that balance is. The urea cycle disorders really underscore the devastating consequences when even one piece of that machinery fails, especially the impact of ammonia on the brain.
Understanding these pathways isn't just academic. It gives you a real appreciation for how life works at a fundamental level, and it connects directly to health, disease, nutrition, and even diagnostics and potential future therapies.
It makes you think about the engineering involved, the constant renewal, the detoxification, the adaptation, like ramping up enzymes during starvation. It's just remarkable.
It truly is a finely tuned system honed over millions of years.
And thinking about the future, we discussed newborn screening, a huge success, and the potential, however distant, of gene editing technologies like CRISPR. It does raise some profound questions, doesn't it?
Such as?
Well, given how precise these metabolic pathways need to be and how severe the consequences are when they're disrupted, as we get better and better tools to potentially modify our fundamental biology, what kind of ethical and societal questions do we need to grapple with?
That's a deep question. Where do we draw the line between treating devastating genetic diseases and potentially enhancing normal function? How do we ensure equitable access to these powerful technologies?
Exactly. How do we balance the incredible promise of eliminating suffering with the immense responsibility that comes with altering the very code that makes us who we are? It's something to really ponder.
Definitely. Understanding the basic science, like the pathways we discussed today, is really the foundation for having those informed, thoughtful discussions about where medicine and technology might take us.
A perfect place to leave it for today. Thanks for navigating that complex world with us.
My pleasure. It's fascinating stuff.
Join us next time on The Deep Dive for another exploration into the science shaping our world.